Okay, hello everyone. Welcome to our webinar today. Today we will talk about ELISA kit again, and today's topic will focus on how to choose the right ELISA kit. So for today's webinar, we will have two parts. The first part will be a very short introduction about the criteria related about the selection of ELISA kit for your experiment. And then the second part will be, I will introduce like how can you tell that whether the ELISA kit, they have a good high performance. And also I will show you some case studies between our CUSABIO and other vendors to show you like how good are our kits. So let's begin the journey today. Here is the 4 criteria that I think important to choose the proper ELISA kit. So basically experimental requirement, brand reputation, and methodology of the ELISA kit. And then the last will be some QC parameters that related with the ELISA kit. The first is the experimental requirement. So I will say that, well, everything should begin with your requirement about the kits. Like you should be sure that which species are you studying and which target analyte you are trying to detect. And also please be sure that you know your sample type, like whether they are the serum sample, plasma sample, or the cell culture supernatant. And also the sample volume will also, like you need to be clear because sometimes the kit, they require the sample volume, like 50 microliter or 100 microliter. So be sure that the kits, they fit your available sample amount. And then the next will be like the detection range. Sometimes the concentration, the analyte concentration of your samples, they may be low or may be high. So be sure that they fit in the detection range of the ELISA kit. Then another thing is the sample size. So confirm the number of the samples that required to finish the experiment. Then based on that, you can select the proper number of the kits to finish your experiment. The last one is related with the experimental environment. I would say that basically you should know the temperature or available instrument in your labs. Then based on that, you can choose the proper ELISA kit. The second criteria I will highlight is like a brand reputation, because sometimes when we buy the product, we care about the user feedbacks, right? Because user feedbacks, they reflect the product quality. Like the user feedbacks can be like it can focus on the manufacturing information and the pre-sale service and after-sale service. And one other important is the literature citation. So based on all this, we can tell that whether there's any potential issues about this product. And so it's kind of like providing a reference for us to choose a proper kit. The third criteria about the selection is the methodology of the ELISA kit. We all know that for the ELISA, they are different types. And for different types, they can be applied in different applications. So here I list the 4 different types of ELISA: direct ELISA, indirect ELISA, sandwich, and competitive. So one example about the sandwich ELISA, the application is different from the competitive ELISA. With the sandwich ELISA, we are able to detect the large molecular antigens or antibodies. But with the competitive ELISA, it's opposite. We usually use this ELISA to detect the small molecular antigens, such as hormones or drugs. So you see, it's really based on your requirement, like which type of ELISA you required to finish your experiment. So based on that, be sure to choose the proper methodology and then the proper ELISA kit. The last criteria I think is really important because it's about the performance of the ELISA kit. And here I list the several parameters that related with the high performance. Like the standard curve, the sensitivity, stability, specificity, and the coefficient of variation. And the last is the operational consideration. So I will talk about these parameters later in details, but I will say all this, they are related with the high performance of this ELISA kit. So here's a summary about the criteria. So I will say always the needs first. Be sure about the purpose of your experiment, then choose the good one. And then take a consideration between the performance and the brand reputation. Then with all these four criteria, I guess you will choose the best one, for your experiment. Then the second part, we will talk about the critical QC parameters related with the high performance of ELISA kit. And then I will show you several cases about the comparison, like between the CUSABIO and our competitors. And the last, there will be some literature cases that showing how our ELISA kit can help the researchers to do their work. Okay, let's start with the criteria, a critical QC parameter. The first one I think is important is to make sure the detection range of the kit. So because, usually we start with the standard to build the standard curve, and the actual detection value of the samples can be calculated based on the standard curve. And then for here, we usually care about the correlation coefficient, because this one we usually require the R to be greater than 0.99. The second QC parameter I think is important is the limit of the detection, or we can say the sensitivity. The sensitivity is usually the lowest the measurable values, and is usually calculated based on the signal of the background and the inherent variability of the assay. So we usually require the sensitivity to be high. And here is a figure summarizing the comparison between CUSABIO and other two brands. And the y-axis is the different target that we made a comparison, and the x-axis is the sensitivity. So as you see, CUSABIO showed a very good performance than the other two brands. And here is a table. So this table summarizing the product from the IS series. So the IS means the industry standard. So all these kits, they are developed specifically for industry customers, so such as the pharmaceutical companies, and they have a very super performance than some other kits. The third QC parameter is the linearity. Linearity, I will say that is the test index of the experimental accuracy and the specificity. So to access the linearity, we usually need to spike the sample with very high concentration to different metrics, and then we will make multiple dilutions with the diluent to perform the assay. So usually measuring the multiple dilutions of the known positive samples by ELISA, we want the concentration is in a linear pattern. So to be simple, so the concentration of the target protein will be determined by multiplying the dilution factor, and then to get the final concentration. So for the best result, we want the concentrations of the sample from different dilution group. Finally, they should be similar for all these dilutions. So here's one example. See, these are different dilutions that we performed, and here are the different the sample metrics. The dilution, after dilution and after we calculate it back, the range is between 80 to 120%. So if they fall in with this range, we will say that all these kits, they have a very good linearity. The third QC parameter is the coefficient of variation. So this one represents the precision of the kit and also the reproducibility of the result within and between the assays. With the CV calculation, it can help us to identify any inconsistency and inaccuracy in the result. We have two different types of CV, the intra-assay CV and the inter-assay CV. So for the intra-assay CV, usually the CV was tested 20 times on one plate. And for the inter-assay is tested in 20 assays. So one is intra-assay CV and the other one is inter. So we always want the CV to be low because the larger the CV value, the greater the inconsistent and error may happen during the experiment. So as you see, for the intra-assay procedure we have here, the CV number is less than 8%. And for the inter-assay CV, the number is less than 10%. So for our CUSABIO kit, we always have this requirement for all the kit. The next QC parameter is the percentage of the recovery. So this parameter is to make sure the assay is ideally for the sample matrix. So how can we calculate the number? It's determined by spiking a known amount of the purified target protein into a sample matrix. And this spike the sample will be measured with the ELISA kit. And the concentration will be calculated from the standard curve. So we want this calculated concentration to compare to the known concentration of the protein. And we want 100% recovery because 100% recovery means that the observed concentration was the same as the actual concentration of the spiked protein in this sample. So the higher, the better. And as you see, we take the ADP, the human ADP as a case, and then we calculate the percentage of the recovery. So our ELISA kit shows like a close to 100% number, and meaning that our kit, for this parameter is very good. The next one I think is related with the stability of the kit. So for the stability, not only the transportation stability, but also like before we transport it, we will perform the accelerated stability test. So this test will test the kit with different temperatures and also test the kit with different days at the same temperature. So all this is to make sure that our product is qualified before we deliver them to you. So it's kind of like a product inspection. And also we will guarantee that during the whole transportation, the kit will be stable. The next one is the specificity. So specificity, to be clear, is to make sure that the kit will specifically detect without the cross-reactivity with some other close-related or the similar molecules. So for example, here, we use the mouse TNF-alpha kit as an example. So we use this kit to measure a panel of different species, like the target protein from different species. And nothing can be identified and detected. So this kit only recognized the nature and recombinant mouse TNF-alpha, and no adverse cross-reactivity was observed. So our kit is pretty specified, has a very good specificity. The last QC parameter is a validation about in a real situation. So we perform the validation of our kit with the nature samples. So to make sure that this kit not only perform well with the controlled conditions, but also accurately and reliably detect the target analyte in a real complex biological samples. So make sure that no contamination and no hemolysis. So from here, I already introduced the 8 critical QC parameters with the high performance. And then I will show you several cases, the comparison cases between us and our competitors. So the first comparison is between CUSABIO and the competitor vendor C. And the kit we used as example is the mouse interleukin-1 beta, the IL-1 beta ELISA kit. So for these two kits, we perform the standard curve build-up. And so the table here is the values, the real values from the standard. And we test the intra-CV first. And as you see, the CUSABIO showed super CV comparing to the vendor C. And also for the vendor C, we observed that for some of the concentration from the low group. So for this one, for the low concentration group, we observed that the OD values, they are pretty close to the black whales. So we believe that this will lead to some inaccurate measurement of some of the samples. And actually it's true. So here we use the same kit, the 1L 1-beta ELISA kit, to measure 8 mouse serial samples. So the same samples were tested with the CUSABIO kit and the vendor C kit. For the CUSABIO kit, the concentration we got is from non-detectable to 60 picogram per ml. And for the vendor C, the concentration range we got were pretty high and large. So it varies from 42 to 225 picogram per ml. We checked the past report, and we found that the concentration with our CUSABIO kit detected were close to the reported value in the literature. And the OD values that the concentration that got from the vendor C kit, the concentration was significantly higher than reported in the literature. So I think we, in this case, I will say that the CUSABIO have a very good performance. The next comparison is with the human interleukin-6, the IL-6 ELISA kit. So with this kit, comparison is also is still between the CUSABIO and the vendor C. So we still start with the standard, and we calculate the CV first. So as you see, CUSABIO still showed the super CV comparing to our competitors. And we also did another comparison to detect the samples. So these two kits, they were used to test the serial samples. So the same samples and two different kits. The result showed that with the CUSABIO, we are able to detect 3 samples. And for vendor C, with this kit, we are only able to detect the 2. So it's kind of like the detection rate of the from the CUSABIO was slightly higher comparing to our competitors. Then the third comparison is with a different kit, the human interfering gamma, the IFN gamma ELISA kit. This comparison was between CUSABIO and the vendor A and the vendor B. So as usual, we start with the standard and build the standard curve. So 3 kits, they were tested for the intra-assay CV. And still, CUSABIO showed super CV than the vendor A and the vendor B. Besides that, we also checked the sensitivity. So here's a summary table summarizing the sensitivities for CUSABIO vendor A and vendor B. We found that the CUSABIO has a relative high sensitivity. So as you see, the sensitivity we have is the 1.95 picogram, and the other 2 kits, they are 3.9 picogram per ml. So CUSABIO ELISA kit has a relative high sensitivity. The last comparison is with the competitor vendor D, and this time we use the human hepatocyte growth factor ELISA kit as an example. And we use these 2 kits to test the supernatant of the PHA-stimulated PBMC cells. And this time we focus on the linearity. So to show you, to introduce again the linearity, we want that the concentration of the samples, they should be similar for all dilutions. So as you see, we'll perform several dilutions for the samples, and the CUSABIO, they have a very good linearity because the concentrations are close to the original undiluted one. But for the vendor B, So you see that this range is a pretty large. So it looks like that the linearity of the vendor D ELISA kit are not as good as the SuperBio shows. So I would say that in this case, the linearity of CUSABIO was super. I already talked about different cases between the CUSABIO and other vendors, but from here, I will show you that even within our CUSABIO, we did the comparison with our kit. So as I mentioned before, we have an IS serials ELISA kit, and this kit is for the industry, is with the industry standard, and has a very good sensitivities. So I will show you several examples about the CUSABIO kit between the IS serials comparing to some other normal kit. This one is the target, measuring the rat TNF alpha. And then we will use this one as example. So as you see, is a summary table summarizing the detection range, sensitivity, and the sample type for these two kits. And we also perform the standard curve assays. So as you see, the detection range, sensitivity, and the sample type for these two kits, they are different. The IS, we have more sample types that we can, perform the assay. But the good thing is that for both of the kit, the linearity, they all between the 80% and to the 120%. So the key point here is that I want to show you that we have a new serials, but and also we can provide you another options if you care about the sample type. So just meaning that you can in CUSABIO, you can always find a good one. Then the next comparison is with the target, the red interleukin-5, the IL-5 ELISA kit. We also have two kits. So one kit from the IS serials and the other one is the normal one. We perform the standard assay. And as you see, the detection range are different. The sensitivity is different. And also the assay time is different. So if you want to perform the ELISA experiment, finish in a short time, then you can choose this IS ELISA kit to perform your experiment. The next one is the human in serine light growth factor 1, the IGF-1 ELISA kit. So for this kit, we are comparing this the OS series with the normal one, the standard assay, and also for these two kits, we perform the assay to evaluate the percentage of the recovery. So the key point here is that the detection range is different and the sample volume of our two kits are different. But the good thing is that the recovery rate for these two kits, they're all between the 80% and 120%. So both of the kits have a very good performance. The last one is the human interleukin-10, the IL-10 ELISA kit. So the same, we have a summarizing table, detection range, sensitivity, and the sample type. And this time, we perform the assessment about the intra-assay precision and inter-assay precision. So for both of the kits, the intra-assay CV is less than 8%, and the inter-assay CV is less than 10%. But based on the kit, the detection range and the sensitivity are different. So if you care about the high sensitivity, you can always choose the IS ELISA kit. Otherwise, the other one will still be a very good option. So hopefully, based on the cases that I showed you, no matter between the CUSABIO and the other vendors or the comparison within the CUSABIO kit, you will find that our kit always show a very good performance. And here is the summary about the hot selling CUSABIO ELISA kit that we have. And the highlighted part in this slide is the citation number. So as you see, we have some ELISA kits that really have a very good, the the good feedback from the customers. So as you see, the TNF, the IL-6, they all have a very good citation numbers. And I think we got really good feedbacks from our customers. So please take a look at this table. And if you find some interesting target, you can always talk to us, and then we can give you more details. The last is some literature cases about how the researchers can use our ELISA kit to do their research. The first one is the publication from the AS Narrow. So this one is about the delivery tour, the efficient nose to brain delivery. So in this paper, the researchers used the 4 different ELISA kits to access the biomarkers and some expressions, the expression of some biomarkers. So CGRP, IL-6, 5-HT, TNF-alpha. So they use this different ELISA kit to compare the expression of this target in the model group and comparing to the control group. So yeah, so I think they've got very good results. And the next cases is about a plant study. So in this paper, the researchers used the ELISA kit, the ABA ELISA kit, to determine the endogenous ABA concentration within different growth stage. And also they are testing the mechanism of the drought stress amelioration with this tool. So I think they have a very good experience with this ABA ELISA kit. Summary again. This slide is about the features and advantage of our ELISA kit. So our kit can be used in different species. We offer a lot of different choice. So multiple species and a large number of the target. And also we have a very good citations record. And we also offer some small trial size, like the 24 trial size for some of the ELISA kit. And also we have the complete and strict QC process about our product. We also offer some the service, like before the sales and after the sales. We also have a 100% risk-free performance guarantee. So making sure that we can solve your problems as quick as possible. Okay, last, please give me several minutes to introduce our company. We are CUSABIO. CUSABIO means customer satisfied biological reagent. And we are specialized in transmembrane protein expression and the antibody screening. So besides the ELISA kit, we offer proteins, antibodies, molecular biology product, and the small molecular antigens. And also, our product has been exported to more than 80 countries, including the Europe, American, Japan, and Germany. Besides that, we have a very good cooperation with famous companies, and our product has been recognized by them. So famous companies like the Pfizer, Moderna, Zinker, Johnson & Johnson. So yeah, as you see, we have a very good relationship. Thank you so much. And here is the LinkedIn barcode. If you want to stay connected with us, please scan this barcode, and we can talk. We can talk more there and have a more communication in the LinkedIn. Okay, so thank you so much. Thank you for your listening.